plaques and clots develop at all, these are the risk factors. Hypertension, cigarette, diabetes, lipids, obesity, alcohol, smoking, atrial fibrillation, prothrombic states, cardiovascular disease, and genetic predisposition. Because a lot of people with all these risk factors don't develop plaques, don't develop stroke. So there's a genetic component. Finally, a disease is a result of an interaction between genetics and environmental risk factors. We have been involved in stroke genetics here in Hyderabad itself. In fact, it was 10 years or 15 years ago when the stroke gene was discovered in Iceland, Antarctica, and they called it STRK1. It was an isolated stroke gene, only stroke gene. You know, even when you remove the influence of hypertension, diabetes, everything else, this was thought to contribute to stroke. But then it could not be replicated in all parts of the world. It could be replicated in Japan and some parts of China. And surprisingly, in Hyderabad, when we did this genetic study, it was an ICMR funded project, we found the same gene here. In fact, this gene consists of a component called phosphodiesterase 4D. On stroke gene, this phosphodiesterase 4D polymorphism, and it was found significantly in the population of Hyderabadi Indians, and it was published. But whatever may be the mechanism, whether it's a large vessel disease, whether it's a embolic disease, whether it's a small vessel disease, the final pathway by which the blood circulation is stopped is thrombosis. And it is this thrombosis which causes stroke, which can happen because of all these mechanisms. And when this thrombosis happens, when this ischemia happens, body tries to fight it by collaterals or by intrinsic thrombolysis. Body produces its own thrombolytic substances. When these things succeed, we call it was a transient attack. We call it transient ischemic attack. And when it fails, we call it a stroke. So basically both are same. Only in one it recovers, in one it doesn't recover. But stroke itself also is not as simple as it seems. It undergoes through a certain process of evolution after the thrombosis occurs. If you look at this, take it from the point 0, that is time 0. At that time, normally the cerebral blood flow is 50 ml per 100 grams per minute. And once it comes below 20, there is a functional impairment. Below 20 ml. Once it comes below 10 ml, there is a membrane failure. And once it comes to 0, there can be death of the cells. But this also happens, there is no immediate death. It happens along the time. So up to 25 or 30 minutes, we can save the brain. It is dysfunctional but not dead. But from 30 minutes onwards, as you can see, the single cell necrosis begins. By 2 hours, almost a lot of it is already damaged. There is a lot of necrosis in this. By 2 hours. And only this much is remaining. And after 3 hours or 4 hours, we are trying to save only this much. What we call penumbra. Therefore, if we have to save stroke patients, we have to act in time. We have to act within first half an hour or 1 hour. Because of the evolution of the acute ischemic stroke. Now, when this stroke happens, when this infarction happens, what happens? It is an interaction between all these substances in the brain, and all of them are important. Initially, we used to think it's only blood vessel. But blood vessel is only one player. The central one is blood vessel. It is surrounded by neuroglia. Glia is thought to be very important, taking part in this stroke damage. Then there are astrocytes. Then there is a matrix. So this is the whole scenario when a stroke happens. As soon as there is ischemia, the first thing which gets activated, surprisingly, is microglial activation. If there was some way by which we could stop this microglial activation, we could stop stroke damage. Till now we don't have anything. We don't even understand glia very well. We thought glia was only glioma, but no. There's a lot in glioma, a lot of activity. The stroke itself is, the whole cascade starts with glial activation. Within 1 day, there is a leukocyte infiltration, there's a lot of inflammation. By 3 days, there is macrophages. And all this is controlled. The response of these leukocytes and microglia and macrophages is controlled by the inbuilt genetic machinery in each and every cell, which you can see on the top side. There are genes, and these are the names of these genes: cytokine, adhesion molecules, and The meaning. When a stroke happens, when ischemia happens, when infarction happens, it's like a full-fledged Mahabharata. There are so many players within it. Big players, small players, elephants, horses, chariots, swords, blood, damage and damage. This is stroke. If you want to know what is stroke, this is stroke. War going on. Now replace them with leukocytes, polymorphs, macrophages, adipocytes, and that is this. Translate that picture into this picture. There is ischemia. There is the exodermal release of glutamate and excitotoxins. Those excitotoxins are just damaging the neurons. There is the floodgates of kinase receptor and AMPA receptor and AMDA receptor open. Sodium flux is in, calcium flux is in. There is the failure of ionic pumps. There is increased cellular sodium, calcium, chloride. There is the stimulation of phospholipids, increased free fatty acids, arachidonic acid. All the free radicals, they are causing all the havoc. And this is the genetic machinery which you can see inside the nucleus. Even the genetic machinery is damaged and attacked by these phospholipids and by these things. So therefore oxygen radicals, mitochondria are damaged. And what is the net result? This is the whole thing is an inflammation going on, stimulated by the ischemia. The net result is injury to the cell membrane. And you see these citicolines and all, they claim that they protect the cell membrane. They are not completely correct, but that's the idea of the cell. But it can't happen. It's not that easy. Because you have to control each and every component, each and every soldier, elephant, horse here. That can't happen. This is again, I'm coming back, I'm showing in between the world literature and with our own work. This work was done in Begumpet, in Institute of Genetics with one of our collaborators, from our stroke patients and published in the International Journal Inflammation by our genetic collaborators. C-reactive protein and nitrous oxide levels in ischemic stroke and its subtypes. Obviously, C-reactive protein is an inflammatory marker. So when you take these stroke patients and take their blood, there are huge quantities of C-reactive protein compared to the control, which only means that the inflammation is taking place. And the more the C-reactive protein, bad the prognosis. And it depletes the tissue of its nutrients. Zinc, very important. People have seen zinc, magnesium, selenium, everything becomes less. This study again was done here. Depletion of serum zinc in ischemic stroke patients. So this is the sequence of molecular events what happens in stroke. Mahabharata lasts for 18 days. This also lasts for 18 days. Starts from zero, ion channel changes, activation of voltage channels, glycine release, glutamate release, nitrous oxide. Nitrous oxide is the villain. It's the worst thing. If we can remove and control nitrous oxide, 80% damage will not happen. But we are not able to do that. So within 3 hours the damage is over. After that the thrombolysis and all is just a formality. It won't happen. It's already damaged. Then next 15 hours there's inflammation for 5 days, free radical generation, and by 15, 16, 17 days the damage is done. Then it's only rehabilitation. So this is the overview of stroke, friends. Stroke pathophysiology. Something which develops over decades. It is not a cerebrovascular accident. The foundations of it are laid from the age of 15 years onwards. So hemodynamic changes happen when the plaque develops. There are hemodynamic changes that happen over decades. Then for years there is a metabolic changes. That means there is less blood going, but it extracts, tissues extract more of oxygen. More of oxygen extraction ratio. Still the patient thinks he's alright, there's no problem. He keeps on drinking, smoking, enjoying. He doesn't know that he's living on the precipice and the edge. And then within hours, he gets the stroke. But there is still a chance of penumbra and all, and within minutes he gets the damage. So this is the whole snapshot scenario of a stroke. Something which develops over 30, 40 years. Then the result is within 1 or 2 minutes, finally. Now we come to biomarkers. Biomarkers which will identify the people who are at risk of getting a stroke so that they can put their act together. And biomarkers after the stroke, which will help us either to diagnose or to treat the patients. Before the stroke is genetic, cardiac, imaging and blood. That's our hope, that if we take these samples, we can identify people who are at risk of stroke. Now, this is again our own work which is already published. These are various gene polymorphisms, and in this, you only have to give a blood sample, and if you find this PDE4D, which is a stroke gene found in Iceland and also found here, then you are at the risk of intracranial disease, extracranial disease, small artery disease. All kinds of strokes can happen if one has this polymorphism. If one has an AS polymorphism, he will get intracranial disease alone. So therefore, each person has a genetic portfolio. So if you know your genetic portfolio, it helps a person to understand how much at risk he is. Then what are the cardiac biomarkers of stroke? Atrial fibrillation is the biggest biomarker. If you know a patient has atrial fibrillation, he is 17 times at risk of getting a stroke compared to a non-atrial patient. And in fact, ECG helps here. I think ECG is a biomarker. Unless clinically sometimes we can miss this patient, clinically we misstated. It looks almost regular. But when you do the ECG, it looks abnormal. Myocardial infarction is a biomarker. Because when you have a myocardial infarction, you have a propensity to develop clots inside the heart. And any patient who has a myocardial infarction, he is at risk of stroke more so in the first 6 weeks, little less in the 6 months, but almost always at some risk throughout his life. Because there are some, the myocardial wall is damaged. Sixth-Sinus syndrome, tachycardia, bradycardia syndrome, if we detect it, it's a marker for stroke. They are at high risk of developing a stroke. And these all things which can be detected by echocardiography are high-risk cardiac sources. Mitral stenosis, prostatic valve, mural thrombus, ventricular aneurysm, echinacea, dilated cardiomyopathy.